So welcome everyone to the talk I'm giving today for the BEAR Conference. This is going to be on a piece of work I did as part of my PhD looking at how blood flows over very small ridges on the coronary artery surface. There we go. So some introduction to who I am at the start. So I do my PhD in Biomedical Engineering, which is in the basement of the Mechanical Engineering building, although obviously we haven't been there for a while. My PhD is mostly based around doing computer models of blood flow. So I've applied this in a couple of different areas, looking at the design of dialysis catheters, which is something that was presented earlier in the conference itself. I've looked at blood clotting in arteries and whereabouts you'd expect that to occur. And the work that I'm going to be presenting today is how the roughness on the surface of an artery can affect the blood flowing through it. So I'm mostly interested in computer simulations because of the lovely images that it makes. So these are just a couple of examples of why you should all love computational fluid dynamics, or CFD, because clearly, as you can see, it's the most objectively attractive science that you can do. So you have some platelet tracking in the left-hand side, so that's looking at areas where blood clots would occur. There's a nice image of turbulence forming around the rotor in the top right, and the bottom image is logo aerodynamics. So that's looking at how the air flows around the BEAR resources logo. So if they want to get in contact with me to improve the aerodynamics of their logo, then my email is at the end of the presentation if they'd like some advice for that. So what is a biomedical engineer? I've written a description at the top, but basically I'd consider it to be taking any of the engineering principles and methods that you learn during whatever undergraduate degree in engineering you do, and looking at applying that to any context that improves any biological aspect, so patient quality of life, be that implants, developing new medical devices, surgical techniques, things like that. So the images at the bottom are applications that our lab has worked on, be it plates for arthritis surgery, spinal implants, or surgical devices for tumors, things like that. Or you could work more on the theoretical side, so trying to improve understanding of human physiology through models or experiments that are difficult to do with clinical patients. So for example, looking at mitral valve functions, blood flow in the atrium, or again, the interaction of blood flow in medical devices. So there's really quite a wide range of things that biomedical engineering encompasses, and that's just some of the ones that we do in our lab in UOB. So today I'm going to be talking about the reasons that we want to model blood flow in the first place. So looking at what can we do with it, how do we do it, are there any reasons why modeling blood is more important than other things? Then we're going to look at why, or what does your artery surface look like? Maybe that's something you've never wondered. Maybe that's something you'll be interested to find out. But what does the artery actually look like if you zoom in very close to it? And then the stages of developing sort of a computational model of an artery. So how does it start? What steps do you need to make? And how do we go from patient data to looking at some hopefully interesting results and conclusions from the model? So we'll get some basics out of the way now. So blood is a mixture of red blood cells floating within plasma. So you can think of blood in a very simple way, which is a red liquid that's kind of like water that flows through your body. And that's one very basic approach to modeling blood. Or you can zoom in on it in more detail and think of blood as this mixture of very small cells floating inside plasma, and these cells can group up in places and they can move around, and that will change the property of blood. Or you could go into even more detail and say, well, there's not just cells floating around, there's proteins, there's other cells, there's nutrients and things like that, all in these different concentrations. So blood, depending on how you want to think of it, can be a very simple fluid that behaves in one way, or you can go into different levels of details of what you think blood actually is and whether or not that's important for the models that you want to make. And then we can simulate blood flow using computational fluid dynamics, or CFD. So the same equations that govern how blood flows through an artery are the same ones that tell you how air is going to flow over a wing or fuel through an engine. So that's why engineering can be applied to all of these different places, but in our case, it's for biomedical purposes. And ultimately, we're trying to improve these biomedical models that we're making with the idea that you move forward towards the most ideal solution, because ultimately we're approximating the body and we want to advance in steps to get towards the most realistic simulations that we can. So the briefest bit of biology that you need to know is that when we talk about, or when I talk about arteries for this presentation, we're talking about the left coronary artery. So that's the one circled, which sits on the outside of the heart. It's one of the bigger arteries in the body. And you can see it branches in multiple places and gets smaller and smaller and smaller. But in this study, we're really looking at the largest part of it just before it branches, which you can see in the right image. So a healthy artery is obviously clear and free of obstructions. And one of the diseases that affects the artery is atherosclerosis, which is the fatty blockage in yellow that you can see blocking the artery. So that's something that a lot of computational models look at. And one of the key parameters that we investigate in terms of the health of the artery is wall shear stress, which is the force that blood exerts on the artery as it flows past it. So this is like the pulling force that blood exerts on the artery. So as the fluid flows over the surface, it tries to pull it along with it in the same way. If you put your hand out of the window of a car and you feel you feel air flowing over the surface of your hand, as it pulls on your skin, that's the pulling force that blood is exerting on the artery. And that's quite important for the health of an artery to have just the right amount of wall shear stress. You don't want too low, that can cause problems, but you obviously don't want a very high wall shear stress, because that will cause the artery to rupture and things like that. So that's most of the basics in terms of biology and what we're trying to do. But why we model blood in general is mostly because simulations can tell you things that you would never be able to see in a patient if they came into a hospital, because the scans, the measurements that you could take of a person might tell you the velocity in some places. You could look at the pressure, how things are changing, but in terms of examining a wide range of parameters that we might want to look at, that's something that you won't be able to measure in a person. So we can learn a lot more about what's going on in terms of the blood flow, and from that can tell us things that we might not have been able to realize, which can inform our knowledge of how vessels work and how diseases begin, get worse, and ultimately progress as things go along. So some examples of applications from these models would be trying to improve medical devices that I spoke about earlier, predicting where these blockages in the arteries are going to form, these plaques are going to form, and even in cases taking a patient's scan of a diseased heart, for example, and getting a surgeon's opinions on what surgeries they might consider for this person, creating a model of each one and assessing the outcomes from each of these surgeries to work out which would be the best one. So that was a case study that wasn't done for a particular patient to choose, but it gives you an idea of, as these progress, they could inform clinical decisions as they build up in complexity and the time that it takes to create these models. Okay, so what does your artery surface actually look like? Well, it's not completely smooth, for one thing. If you zoom in close enough, you start to see in these bottom images, it's covered in small ridges in kind of a swirling pattern as it flows through the artery. So kind of in the same way a gun barrel might be rifled to smooth the flow of air over the bullet as it comes out, the artery surface is covered in these very small ridges as it flows along. You can kind of imagine them as if you zoomed in on the tip of your finger, those ridges that make up your fingerprint as you get very close would be the texture on the artery itself. Although obviously these ridges are very, very small compared to the size of the artery. So a coronary artery would be 3 or 4-ish millimeters in diameter, and these ridges themselves are 4 or 8 microns high. So relative to the size of the artery, they're much, much smaller, and that's why a lot of people consider it to be a smooth wall when it comes to doing these kinds of models. But why do we care about how rough an artery surface is? Well, for one reason, there's a sort of healthy level of roughness for an artery surface, and the roughness of that surface increasing dramatically is something that can be an early sign of atherosclerosis that I mentioned earlier. Those blockages in the artery as fat builds up on the surface and underneath the surface of the artery, it inflames the surface and begins to get a lot rougher. So that's one of the early signs, but that's something that is actually very challenging to measure in a patient if they were to come in, because whilst it does increase, this is from around 10 microns up to 50 or 100 microns, which is still very, very small compared to the size of the artery, and would be difficult for you to see that with ultrasound. So whilst we know about it, it's not a useful thing to be able to measure yet with the current things that we have. So it's clearly having an impact or is related to the health of the artery, but it's something that hasn't yet been studied in these computational models, because they typically focus on a bigger scale, you know, what's going on in a branch, what's going on with a blockage, things like this. You don't tend to include these very small effects on these models, but obviously they're playing a role. So that's kind of the two parts of where the motivation for this project comes from. Is one, what effect does this roughness have on the blood flow, on the health of the wall in terms of the parameters that we'd usually investigate? How is it affecting the wall, a rough wall that we have measured versus an assumed completely smooth, nice wall? And then the second question that comes from that is, we have several approaches of how we model blood flow. Which of these is going to be the best for modeling blood on the big scale in terms of flowing through the artery, and the small scale flowing over this very small surface? So we need a model that's robust and can do both of these very well. And we're going to look at different approaches of how one model performs versus the other. So those are our two ideas behind this kind of model. So these are the sort of general steps that you would go through in making really any cardiovascular model. So to begin with, you would need your geometry, which is your shape of the artery, your size, your length, your dimensions, things like this, as well as the roughness of the surface. We would then take that and split it up into very, very small pieces called a mesh, which is where we solve our equations for what the blood is going to do. So we need to go from a raw geometry into segmenting that into small pieces that we're going to solve our equations in. And then we need some idea of what's coming into the artery and what's going out of the artery. So we're going to tell it that the velocity is varying. And then we need to say, we have a fluid, in our case blood, how does it behave in terms of the simple or difficult approaches for blood modeling that you want to use. So we give it some equations that define what blood is, which we bundle all of that information together and pass it to the solver and give that things like how long do we want to run this simulation for, 5 seconds, 10 seconds, artery time, simulation time, which we pass over to BlueBear, who very kindly provide their resources to solve all of this, and hopefully at the end get out some nice and interesting results of what the blood is doing over the surface, some pretty pictures, things like that. So that's hopefully the plan of making one of these models that I'm going to walk you through now. So when it comes to reconstructing a rough artery, the first thing we need is an artery to begin with. So in these circumstances, we use porcine arteries or pig hearts. And you can see the coronary artery sitting on the surface of the heart there, ready to be extracted. From that, we take a very small piece from the top of the artery, as I mentioned earlier in that circle. which we open up and ready to look at under a microscope. So this is our representative piece of the coronary artery surface, which we're then going to look at under a microscope to get a very detailed picture of the surface, which can then give us the surface of the artery itself. So this is an image which is made not only just taking a photo of the artery, but it gives you 3D coordinates of the surface. So it's millions and millions of tiny points on the surface used to create this image. So this is not just a photo, this is 3D point data used to make the surface, but you can see the ridges forming on it quite nicely there. Unfortunately for us, this kind of information is far too detailed to end up putting in a model. It contains a huge amount of data on the surface itself, and whilst it's very exact, it contains a lot of small artifacts and ridges that aren't realistic on the surface itself, that come up from problems with the imaging and just bits of dust and things like that that have accumulated on the sample. So we need to clean this up before we can implement it into a computational model. So the way we decided to do this was look along these two lines. So if you draw a line along the artery and draw a line across the artery, you can look at how the height varies over those two lines, which give you an idea, a representation of how your artery surface is going to vary. So we have these two profiles that we can put perpendicular to each other, and if you extrude them along each other, you end up with quite a nice realistic approximation of a rough surface of your coronary artery. So this is still a very small height roughness in a small segment of the artery. So this isn't across the entire sort of circumference around the artery. This is just a small section at the bottom that we've managed to create, which bears a pretty good resemblance to the ridges that you see over the artery itself. So now we have our rough surface. We implement that using the data from literature in terms of what size should the artery be, things like that. And we put the rough surface in blue along the bottom of the artery with so the blue surface is the rough part, and the rest of the artery we're assuming is smooth, like all of the other models. So we're trying to compare within one model, how does blood flow over the blue rough section versus the smooth section at the top? What differences can we see between the two? And because this is the first time we're looking at the roughness, we need to put it in the simplest possible case. So at the top of the coronary artery, where it's nice and straight, so we can see differences between the two techniques without having to rule out effects from curvature or a blockage or things like this. We need a simple test case to see the differences between the two walls there. So now we have our geometry sorted. The next thing we have to do is to break that down into a mesh. So a mesh is our way of solving the actual problem. So at each element in this mesh, each of these small triangles you can see at the bottom, we're going to solve our equations that say what blood is doing at each of those points in the mesh. But unfortunately, because the size of this roughness is so small, we need a lot of very, very small elements to make it a nice smooth curve. Otherwise, we'll turn our surface into a very jagged and rough, too rough surface for what we want to do. So we have to represent it with a lot of small elements. So unfortunately, our 8mm long artery is some 65 million elements required to make just a small section of this artery rough. So this is still quite a very, very large number of elements that we have to solve all of our equations in at every single time step. So this is a very computationally intensive process. So we couldn't make an artery with a completely rough 360-degree rough wall with this, because it would simply be too much to handle. So we have now our mesh of the geometry, and then we need to give it some information of what's coming into the artery. So in this case, you can see the velocity waveform. That's how velocity is varying in a typical coronary artery, which is what we get from the literature, and we apply it to the inlet, as you can see in the bottom image. So now we have those things sorted, we can start to think about our different ways that we're going to model blood in this simulation. So on the left in the green, we have our single phase model, which is our very simple approach to blood modeling, where we say blood is a liquid made of one thing, which is very similar to water and has a constant density and either a viscosity, like a thickness, if you like, of blood that is constant or can vary slightly due to shear, which I'll explain a little bit later. So the green is our very simple approach to blood modeling, which is very quick to solve, but doesn't necessarily capture all of what's going on. So on the right-hand side in the purple, you have our multi-phase models, which is where we have more than one phase. So in our case, blood is made of plasma and red blood cells. So we're able to simulate the concentration of red blood cells, how they move through the artery, are there any places that they build up and things like this. And red blood cells obviously have a very varying viscosity and have a lot more complications in terms of how you model a very small cell as opposed to just assuming blood is one bulk fluid. So there's two different approaches to modeling blood that we're going to look at in this one. So we have a graph here of the viscosity of blood at different shear rates for our two approaches. The green is our simple approach and the purple is our complicated approach. So shear is the rate at which velocity is changing close to the wall. So in the image in the top left, the velocity of blood at the wall on the black boundary is 0. Blood isn't flowing at the wall. And just next to that, just above the wall, if there's a very high velocity of blood, then that's a very high shear rate, because as you're moving away from the artery, that velocity changes very quickly. Or in the center of the artery, the velocity isn't really changing as quickly, so there's a lower shear rate. And blood's viscosity will change depending on the shear you apply to it. So its viscosity isn't constant like water's is. It can change over different shear rates. So low shear, the blood starts to become thicker as it moves slower near this wall. It becomes a thicker sort of liquid. And in the high shear environments, blood becomes very thin and flows a lot easier. So low viscosity is kind of thick in this circumstance. Wait, no. High viscosity, like 1, 2, 3, numbers like that, is kind of like honey. And low viscosity, 0.000 something is very low and like air, basically. So we have our two different ways to think about blood in this circumstance. And then the added complication, this is just our multi-phase models where you have red blood cells and plasma. The concentration of red blood cells really affects the blood properties as well. So in a value of 0.2, that is 20% red blood cells. So most of it is plasma, and that has quite a different property than a very high concentration of red blood cells. So when you get above a hematocrit, which is the concentration of red blood cells, of 0.95, blood is no longer a fluid at that point because you have such a large concentration of cells in one place that blood no longer flows through an artery. It will completely just build up and stick in one place. So the concentration that we can see in this kind of model has a really big impact on how blood flows. And then we take, as the next step, all of that information that we have and we take it to our solver, which is going to do all of the annoying maths for us. So in our case, we're going to simulate 7 heartbeat cycles of that velocity waveform that you saw to try and allow completely physiological flow to develop. So we're going to give it a lot of cycles to hopefully establish what will be normal heartbeat rhythm. And we're going to solve our mesh and with our equations as we described before. So we've kind of moved through our plan reasonably well, hasn't taken us too long, and now we're ready to pass over our solution to BlueBear. Except it doesn't always go completely to plan like that, unfortunately. A lot of times making these models causes a lot of errors, a lot of headaches. And before we get onto the BlueBear solution, I'll take you through a couple of the very embarrassing ones that I made whilst making these models. So the first mistake, having just started to use BlueBear, is accidentally typing a command similar to the one you can see there. which unfortunately deletes all of those files, those geometries, those meshes, those boundary conditions. It deletes all of them that you've spent probably weeks trying to create. And in a single enter, you delete every single file in your folder, which is unfortunate, having spent a long time making these models. So we learned to be very patient from that one. And then the second mistake I made was trying to be very organized in how I made these models after that first one. And you solve a model, you build a model and you pass it over to the BEAR team to solve for you, which is very nice. And you sit in a queue for several days waiting for your model to be solved. And it instantly starts to solve and comes back with an error and crashes, telling you it can't find the file at the bottom because you forgot to rename your files. So this teaches you to be very planning orientated, to check your work and submit it in a proper process. But these models can go right and they can go wrong. And it seemed fine to dedicate some time to saying it doesn't always work first time, and there's a lot of headaches that go into making all of these models. So it doesn't always work first time for everyone. But anyway, if it does work, you can submit your jobs onto the Blue Bear resources. So this is probably where I should thank the BEAR team for letting all of this research be possible by providing their computational stuff. So these models with 65 million elements and equations to solve in all of them take a very long time. The multi-phase ones take a huge amount more time than the single phase ones. So there's a disadvantage already. And as you can see, the energy used to make all of these simulations could have been provided to a polar bear for 20 days. So you'll have to be the judge of whether or not this project was actually worth it. So after all of that, we can get to our results for what our rough artery is actually doing. And hopefully there's some interesting differences between roughness and a smooth artery. Otherwise this wasn't worthwhile. So you can see in 6A, the figure in the top right, the difference between a rough artery and a smooth artery. So the smooth is inlaid in the box in the top left. And the first thing you can hopefully see is that this wall shear stress, the force that blood exerts on the artery, is varying a lot over this rough surface. At sort of the peaks at the top of the roughness, the wall shear stress is quite high. And right at the bottom of these dips, the wall shear stress is much, much lower. Whereas in the image just below the 6A, it's constant. There's no variation over the surface at all. It's very, just no variation at all. Whereas on the rough surface, this parameter varies a lot, which is interesting, right? Because you wouldn't, ultimately you're trying to model the same thing. You're saying this is an artery, one says it's smooth, one says it's rough, and you see this difference in your approach. Now, the average over the smooth and over the rough surfaces are actually very similar, which is, again, what you would hope to see. But this difference in wall shear stress is interesting because a value sort of below 1 indicates areas where the artery is going to start to become unhealthy and dysfunctional. And you can see in the dip right in the central channel in the top image, there's a very low region of wall shear stress there, which is a problem even in this relatively healthy, straight section of artery with no reason for any problems, that if that dip started to grow even slightly, if the roughness were to increase there, then that would be one of the early places where you might start to see the artery health declining and either fats or platelets or particles starting to accumulate right in this dip of the artery is one of the places where a plaque or a blockage would start to form in these dips, these valleys of the roughness. And then the bottom image is showing a measure called the relative residence time, which is just an indication of how long a lump of blood spends in any particular region. So near the top, near the valleys, there's a very low residence time. Blood is flowing over the top of the surface and isn't spending a very long time there. However, in the bottom of these valleys, again, you can see quite a high residence time occurring, which is sort of, again, reinforcing that blood is spending longer in these places. Maybe oxygen and other nutrients aren't reaching the surface of the artery very well because it doesn't receive that much blood flow. So again, highlighting that even in a straight piece of artery, there are considerations that you wouldn't have got to with a smooth walled assumption, which again predicts a very uniform residence time. So these are some of the differences that we were able to see between the two assumptions straight away. And then we can look at how this varies over time during a cardiac cycle between the smooth wall and the rough wall. So the rough wall is the dotted line and the smooth wall is the solid line. So the first thing you see is that the wall shear stress on the smooth wall varies almost exactly with velocity, whereas the rough wall seems to be much less affected by that change. So the changes over the rough wall are slower. It's more consistent and stable over the artery compared to the smooth wall, which has quite a large variation. Although again, the averages between the rough and the smooth were similar. Then in terms of our approaches to modeling blood, our simple single phase model is actually in quite close agreement, at least on the smooth wall, during the entire cycle. So there isn't a big difference between our two techniques here, probably showing that the single phase model is doing a pretty good job given how simple it is. But on the rough wall, there is a bigger difference, especially towards the end of the cycle, where the importance of considering blood to be a mixture of cells and plasma and things like that is having a bigger impact. And this is likely due to the rough wall having quite a big variation in the concentration of red blood cells, which is what you can see in this image here. So the image at the bottom is the hematocrit, which is the concentration of red blood cells on the surface of the artery. So a very low hematocrit means there's not many red blood cells, and a very high hematocrit means there's a large number, especially once you get above 0.8 and this starts to increase. This is where blood is not clotting, but this is where large groups of red blood cells are accumulating on the surface. And this varies a huge amount, as you can see, over the surface itself. Whereas in the smooth wall, again, in the top left, it hovers around the sort of 45%-ish region, which is what you would expect in a healthy human artery. So there's unfortunately very little data on what the distribution of hematocrit would be over the rough artery surface in general. There's no real information on the distribution other than 45% if you were to take a blood test is about right. But what we can see in the bottom image is it appears that there are regions where it's a very high and very low, but is the roughness dictating what this hematocrit is on the surface of the artery? So we can plot graphs of what this looks like. So in the top image, you have the dotted line is the height of the artery, and the red is the purple is our hematocrit. And it does look like there are places where the roughness peaks, and so does the hematocrit. And these areas would be, again, susceptible to malfunction because you don't want a very high concentration of hematocrit because blood doesn't flow very well in these circumstances. It becomes a lot more viscous and thick and exerts a much higher wall shear stress as well because the viscosity increases, and that increases the force that blood is exerting on the artery. So it looks like there's some correlation between these two things if you look at the bottom graph, but it's difficult to say whether or not the height is impacting this hematocrit distribution very well. There's obviously some trend between them, but it looks like there's a large number of factors that are impacting this concentration of hematocrit, which is unique to be able to be seen in the multi-phase models, whereas our single phase simple approaches have no way of determining what the hematocrit would be. So this is another big advantage of these complicated multi-phase models, is seeing this distribution and seeing how this changes is really going to affect the simulation, but also the predictions of the model in terms of what's going on in the surface of the artery. But it's difficult to say just from this what the links between the two are. So that was all of the kind of notable or interesting results that I could put together for this. But what does that tell us given our motivations? Well, there's quite a large difference between the two assumptions, our rough wall and our smooth wall. despite the fact that on average things were similar, which was quite promising, you could see quite a big variation in these parameters that we associate with atherosclerosis and disease and things like this. There was quite a large variation over that rough surface, which you were missing out on if you used a smooth wall assumption. And then the multi-phase models, whilst similar to the single phase, in our case on that rough wall, gave a lot more detail and a lot more interesting parameters that you could investigate and different predictions of what was going on compared to our simple single phase model that likely sort of oversimplified what was going on near the wall because it couldn't simply handle these very small changes in geometry, whereas an approach that's based on individual red blood cells, in this case, was able to see more of what was going on near the wall. So any sort of models that people are making, which is really interested in what goes on near the wall of an artery, not necessarily the coronary, might find it interesting to see, just as a comparison, what happens if you try to include some roughness on the surface itself. So there are devices which are called stents, which hold the surface of the artery open like that in cases where people have blockages to try and keep blood flow as high as possible. And these devices are 50 or 100 microns high, whereas the roughness is 4 to 8 microns high, which isn't a massive difference. That's going to have some impact of how the stent holds the artery open. But the simulations and the design work people do to make these stents are based on a very completely smooth artery surface. So that would be interesting to include and look at. And one of the impacts that roughness might have once you start applying it to different cases besides this simple cylindrical case. So as I just said, applying this to different diseased arteries might be very interesting to see this progression, as well as if we started to increase this level of roughness that we were simulating, do we start to notice more or exaggerated things like wall shear stress and residence time in these places? And will that affect our simulations and predictions of where plaques form and how they form as this roughness does tend to increase? As well as, so this was the roughness of the coronary artery, but there are lots of other arteries of different sizes and veins and capillaries inside the body, which will have a roughness as well. And there's a whole branch of these simulations which look at different things, which again, could potentially have some interesting results if you start to consider what this microscopic surface texture kind of looks like as well. So that's kind of the overall outcomes and results of the project that I talked about. If you want to get any more information or talk about them or Blue Bear or anything, you can email me, especially about logo aerodynamics. That's a key one, a new field I'm trying to start. Or if you want to read the paper with some more detail, there's links to it there as well. I guess this would be a good time to thank the BEAR team for putting on the conference and letting us present our work, as well as providing the pretty great service they do for free, to be honest. So thanks to everyone at the BEAR team for putting all of this on and for providing their resources, and especially Runzhao Li for hosting this conference as well. It kind of been easy to put a conference on during a lockdown, so not a lockdown. Thanks for that. And I guess that's basically all I have for the presentation. So if anyone has any questions on all of that, I'll be happy to try and answer them if anyone found any of that interesting.
Yeah, thank you, Dave. Thank you for your excellent presentation. Our attendees, do you have any question? Please raise your hand or type in the chat box. Yeah, maybe Dave, I can first answer a question. Since you are simulating the blood flow in the artery, how do you make the model validation? And since you are simulating some organic in the human, do you have any measures to complete the step of the model validation? And another question is that in order to improve the predictive accuracy of your model, which measures you adopted in your simulation process? Thank you.
Okay, so the validation of these kinds of models is always the tough bit at the end, because whilst it's advantageous for these models to be able to look at new things that couldn't be seen inside the body, it then becomes a challenge to show whether or not these new predictions are correct, because there obviously isn't much information out there to compare it to. So typically, the validation of these models has to be done based on the data that we do have available to us, which there is a decent amount on, and we look for sort of clinical studies on a large number of patients to give us this kind of data. So there are big studies usually done at a national level that give information on general health of arteries, typical values that you would expect in terms of velocity, pressure, wall shear stress in particular. There are lots of well-documented studies that provide sort of healthy ranges and estimates based on these things, as well as your comparisons to other computational models that have similar ideas to you. You can sort of lump all of this together to show that your model is correct, but validation, unfortunately, is one of the more challenging parts, especially on the more novel side of developing these kinds of things. Because as well, it's not something you can experimentally validate very easily either. So you have a challenge that way as well. What was the second part of the question? Sorry.
I think now we have a more professional question. I think you can answer Diana first. Yeah, because yeah, Diana. I didn't find you in the.
I can't see the chat for some reason. Hold
on. Wait a minute. I unmute Diana. Yeah, Diana, I already unmute you.
Hi.
Yeah.
Hi.
Hello. First of all, awesome presentation. Congratulations. And I just had a curiosity. Have you thought of putting together a rough artery model using FSI? I mean, obviously the complexity would be huge, but FSI would have an impact on the flow parameters. It's just a curiosity. I know it's not very easily achievable, but I just wanted to know what you thought, basically.
No, that's very interesting. So I'd say one limitation of all of the work I spoke about is one that a lot of these models have, which is we don't model the expansion and contraction of the artery during its simulation, mostly because the effects that we want to look at will be there with or without FSI. They might change, but it's a limitation that a lot of models have, and we have to accept in some cases. So FSI is the interaction between blood flow and the artery itself. So you look at if blood flows, an artery expands, and then it slows down and the artery constricts, things like this. That's obviously really advantageous in terms of the accuracy of your simulation. But again, it adds another layer of complexity to the model. So in this case, you have to work out that interaction between the blood and the artery at every place that they're in contact. And as unfortunately the rough surface requires such a huge number of elements to begin with, it would take more computational power than not just BEAR has, but pretty much a lot of people have to manage to get that to work. It would be very interesting to see how these things change if the artery was able to deform around the blood flow itself. But again, you would get into limitations of the scale, the very small size of the roughness. It becomes difficult to give it accurate sort of artery properties. You can say that artery is sort of elastic and give it some values for that. But to say the very small ridges are elastic in the same way as the entire artery itself, again, no matter what stage you consider it at, you're going to introduce some simplifications there. But definitely FSI is very interesting, and I'm sure would be nice to include in something like this. It might have to be done not with a full artery length, maybe just a very small section with roughness to try and simplify it all down. But that's something that would be very interesting to do, yeah.
All right. That makes sense. Thank
Cool.
you.
Thank you for the question.
Any more questions from our attendees? Please raise your hand or put in the chat box. Yeah. Oh, yeah. Okay. We receive a new question from Aslam. I unmute Aslam. Yeah.
So the artery was made from the rough segments were 0.8 millimeters long, and the artery was made from 10 repeating segments. So the total length of the artery was only 8 millimeters, which is quite short for one of these models. But the coronary artery is shaped like that for I think a distance longer than 8 millimeters, but then branches, and each of these branches gets smaller and smaller. So our model was focusing right on the biggest, sort of simplest point from then on. But yeah, the 65 million elements were for a very small section of roughness in an artery that was only 8 millimeters long. So that's a very large number of elements for a very small distance. But that's just because, again, the size of the roughness is so small compared to the artery. You really have to have a very small element size so that the artery is smooth, or the roughness is smoothly curved over the surface as opposed to jagged and whatever. So it's a lot of resources, and thanks for setting them up for us.
Any new question from our attendees? If there is no more question, we we got a new question from Debbie Carter. Debbie, I already unmute you. You can directly talk to Dave.
Hi Dave, thanks for that. It was interesting. I did miss the beginning of your presentation, but I thought it was quite brave of you to share the coding mistakes that you made. And I think that's something that interests me, is that people are subject experts, but they're certainly by no means computer and coding experts, so that there is that learning curve for everyone. So I just wondered if you had any advice for people that were sort of starting out with the computational side of things, and any tips or things that you sort of learned along the way that might help people that are new to this sort of thing.
Oh, that's tough. That's the hardest question. There's a lot of things. I guess it's like learning anything else, that it will be frustrating at the start and you will make these mistakes. I'm not sure if like I have any useful or good advice for those kind of things. It would be always start doing the simplest thing you can, be it making a model or any kind of code or anything like that. Would always be start off with the most, whatever your idea or goal is, try and reduce it to what the most simplest version of that is, even if it doesn't even seem relevant or useful to what you want to do in the end. If you can make it very simple and very straightforward to the point where it starts working, it does what you want it to, you don't just get error messages, it gives you something, then already you've learned probably how to do it, but how it can go wrong. And from there you can slowly change things and increase things to get towards what you want as your end goal. Never jump headfirst into something and sit in errors and mistakes and getting nowhere without understanding why. If you can go for a simple version, hopefully get that to do something, and slowly move on from there to improve it over time. But I don't know how much that applies to learning BEAR and coding and all of that. It would just be accept making mistakes and try not to give up when they happen.